So, sa midterms, we started biochemistry. This is again under area 2 of the board exam. And this is considered the hardest area and one of the hardest subjects sa board exam. So you really need to give more time for this subject. We had started with our review with the different organelles of our cells. And then I think, yes, we started our discussion with proteins already. Pero ano pa lang ito? Introduction to proteins. So classifications lang muna kung ano ba yung function nila. Either they are structural or dynamic protein. And then we continue with the nucleic acids. Nag-jump tayo sa nucleic acids since before ang protein ma-produce, nucleic acids muna. I hope na master nyo na yan. Our building blocks for our nucleic acids are the nucleotides. And when you say nucleotides, tatlo yan. You have the phosphate, the nitrogenous bases, and the sugar. So we have two, DNA and RNA. Their difference is on the sugar. For RNA, we have ribose, and then deoxyribose for the DNA. And then we have nitrogenous bases that are unique for the two. Sa RNA, uracil yung meron, and then wala yun siya sa DNA. And then for the DNA, thymine naman ang meron, and then again, wala din yun siya sa RNA. So take note also of the different bonds. Kasi tatlong parts yung ating nucleotides. So do not forget the bonds, the glycosidic, which connect the pentose or the sugar to the nitrogenous base. And then the phosphodiester bond, which is the major bond that connects the backbone of the nucleotide. When you say backbone, it's the pentose or the sugar and the phosphate group. And then lastly, we have the hydrogen bonds that connect the nitrogenous bases. So yung number of hydrogen bond is dependent kung anong nitrogenous base yung i-connect natin. And not forget, we have 3 bonds between guanine and cytosine. Silang dalawa talaga ang partner. And then 2 bonds between adenine and thymine if that is a DNA. And 2 bonds also for adenine and uracil if RNA naman. Okay? And then again, we ended here with the concepts. When you say anti-parallelism, di ba double helix yung DNA natin? The first strand will start at 5' to 3'. So 5' siya mag-start. The 5' is referring to the number of carbon number 5 siya, ang connection between the phosphate and the sugar, and then it will end at 3'. And then the second strand for the double helix will start at 3'. So baliktad, anti-parallelism. And then when we say complementarity, this refers to the pairing of our nitrogenous bases. It is always purine and pyrimidine. So hindi pwede mag-purine and purine or pyrimidine and pyrimidine. Dapat complementary sila dalawa, purine and pyrimidine. And then equivalent pairs, syempre since they exhibit complementarity, kung gaano karami yung purine, ganun din dapat karami yung pyrimidine. So let's talk about the central dogma of molecular biology. That is the reason why we skipped discussing the protein first and nag-jump tayo with our nucleic acids. Kasi ito siya. Magsa-start muna sa DNA, and then DNA will undergo replication to have more DNA, and then the DNA will undergo transcription to produce the RNA inside our body, and then from the RNA there will be the so-called translation, and that's the time protein will be produced. Without protein, there is no cell, and then there is no life. Okay, so minsan tinatawag siyang central dogma of life. This is our process. So again, we have here the DNA that can undergo replication to have more copies of our genetic material. And then, again, mag-undergo siya ng transcription to produce the RNA. Though RNA is also capable of producing or copying its own strand, we also call that RNA replication. And then next, we have the translation, that is the production of our proteins. Now, as you can see, we have a process that directly translate DNA to protein. We call that direct translation. From DNA, magiging protein na siya, hindi mag-undergo ng transcription. Translation na siya agad. And aside from that, we also have the process called reverse transcription. Kasi supposedly it is supposed to be DNA to RNA, but this time, from RNA, gagawa siya ng DNA. Reverse transcription. And this process is, ang nag-exhibit nito is yung HIV. Yung human immunodeficiency virus, the HIV, can do reverse transcription. Since HIV is an RNA virus, so kapag nakapasok siya sa body, re-release niya yung RNA, and then since capable siya of doing reverse transcription, ang RNA niya gagawin niyang DNA. And yung DNA na na-produce will be incorporated in our own DNA. That's the process of being infected with the HIV. And if it becomes terminal, then that's what we call AIDS. Okay? Sige, let us have first a video sa RNA, sa ano muna tayo, DNA replication. Okay, so again, let's have the DNA replication. Our template here is the double-stranded DNA. And then the process is, kailangan mag-unwind muna yung double-stranded DNA for us to have the replication. Link ng video ay paki-search daw sa YouTube kay downloaded man gud ako to. Dili ko kahinumdum unsa ang title ato. So naa siya daan diri sa ako ang laptop. Okay? Again, we have here the enzymes. The first enzyme involved in the DNA replication is our helicase. So ang trabaho niya is to unwind the double-stranded DNA so that the nitrogenous bases inside will be exposed. Yun kasi yung kokopyahin ang nitrogenous bases inside the double-stranded DNA. Okay, that's the first enzyme. The second one is the primase, which creates a primer. So when you say primer, it's actually a small or a short DNA strand that will be used as a starter. So that will signal the start of the replication process. And then we have the topoisomerase. Ito yung ano siya ang kontrabida. So 'di ba, di-unwind nato ang double-stranded DNA. However, malaki ang tendency kasi na mag-coil sila ulit. Mabubuo ulit ang double-stranded DNA. So ano, ang affinity kasi nila with each other, grabe. So magko-coil sila ulit. Now, pag nag-coil kasi ulit yung double strand or dalawang strand ng DNA natin, walang replication na mangyayari. So, to inhibit the supercoiling or the recoiling of the two strands, yan yung work ni topoisomerase ngayon. Pipigilan niyang mag-recoil. Okay? Wait lang. Kita. And then, for bacteria, we have actually the topoisomerase 2, or we call that the DNAligase. That's for the bacteria, ha. And ito yung target ng ating antibacterial agents, yung mga fluoroquinolones natin. Like example, we have the ciprofloxacin, the levofloxacin. They all target the DNAligase. So meaning, yung mga antibacterial na yan, the target is to inhibit the DNA replication of the bacteria causing the infection. Okay? Again, we call that DNAligase. Take note, that's the mechanism of action of our antibacterial. We don't call it antibiotic ha. Kasi antibiotic, remember, paulit-ulit ko na yan sinasabi, not all antibacterial agents are called antibiotic. We just call it antibiotic if it is coming from or it is derived from other microorganisms. This time, since fluoroquinolones are synthesized in the laboratory, synthetic agents sila, we'll just call them antibacterial. Okay? That's the third one. Since topoisomerase yung sa atin, sa bacteria, topoisomerase 2 or DNAligase. And then the major enzyme for DNA replication is the DNA polymerase. Actually, are DNA polymerases. Marami kasi tayong types of DNA polymerases. So sila yung responsible in the synthesis of the new DNA molecules. So take note of the types of DNA polymerases. We have 1, 2, and 3. And then take note of their activities. We have here 5'3" polymerase activity. Ito yung maggagawa na talaga ng new strand. Remember, nag-separate ang dalawang strand. Parent strand ang tawag natin sa kanila. And then kokopyahin yung parent strand, gagawa ng 2 new daughter strands. So ang trabaho na yon is 5'3" polymerase activity. So all types of DNA polymerases are capable of doing that. Kaya nilang lahat mag-replicate talaga ng strands, new strands. But dito sila nagkakaiba sa exonuclease activity actually. Yung proofreading ability kaya ng tatlo, 1, 2, and 3. When you say proofreading, di ba nag-copy ang parent strand. Now, to avoid mutation, since kapag hindi perfect ang pag-copy ng parent DNA strand, mutation ang tawag natin dyan, kailangan niyang balikan yon, yung sariling trabaho niya, balikan niya yon to see or to check if tama ba talaga ang nakopy. So, actually mas marami ang mutation sa RNA kaysa sa DNA because the DNA polymerases are actually capable of doing proofreading. So, all of them, 1, 2, and 3. Pero yung removal of the RNA primer, only the DNA polymerase 1 is capable of doing that. That's still, that's baliktad, 5'3" exonuclease activity. Babalik siya. Diba 5'3" yung strand natin, yung kabila 3'5". So yung DNA polymerases 1 lang yung capable of doing the removal of the primer from the 5' to the 3' end of the strand. Okay? So, take note, DNA polymerase 3, ito talaga yung nagsisynthesize ng DNA. It's actually the fastest enzyme that will do the DNA replication. So, the DNA replication happens from 5' to 3', and then the proofreading baliktad. Siyempre, babalikan niya yung trabaho niya. That's 3' to 5'. But again, DNA polymerase 1 is the only DNA polymerase that is capable of removing the primer. Okay? But this is also required for proofreading. Kaya niyang mag-repair. So that is why again, I told you a while ago na mas marami yung mutation sa RNA compared to the DNA. Sa RNA kasi hindi talaga siya nagpo-proofread. Okay? And then, lastly, we have the last enzyme here, the DNAligase. So, anong trabaho ng enzyme na ito? This will join the Okazaki fragments or the fragments of the lagging strand. Okay? So, kasi, ang mangyayari, ganito. Diba, we have the double helix, nag-uncoil na sila. Titigilan na yan ng topoisomerase. If this is the parent strand number 1, ayan, magre-replicate siya. Pag tuloy-tuloy ang replication, we will call it the leading strand. Okay? Pero itong isa kasi na strand na nahiwalay, hindi kasi tuloy-tuloy ang kanyang replication. By fragments siya. Ang tawag sa isang fragment na yan is the Okazaki fragment. And then afterwards, after makopy na lahat, all these fragments will be joined by, ililink sila together by the enzyme called DNAligase. So yan lahat yung mga enzymes in our body na involved in DNA replication. Questions? For ta mag-end? Wala. So, ano yung tinatawag natin na genes? When you say genes, these are the fragments of the DNA. Yung mga small pieces of DNA natin sa isang strand, yan yung tinatawag na genes. And dyan, class na encode lahat ng ating characteristics. Okay? The genotype and the phenotype. When you say, I think I have the genotype and the phenotype here. Right now. Let me check. Yeah. When you say phenotype, that's the physical traits and the appearance. Makikita natin siya physically. Pero na-encode yan lahat sa ating genes. And then yung mismong sequence of the genome is called the genotype. So not all na na-encode dyan sa sequence na yan can be seen in our physical trait or ang appearance natin. Yung mga recessive genes, andyan pa rin sa atin and we can pass it on sa next generation. So pwedeng doon sa kanila lumabas yung trait na yan. Okay? So next meeting, we will continue with the RNA polymerase and the translation process, and we will also discuss the reverse transcription. So by the end of that, we will have the protein. So after that, syempre isa-isahin na natin yung mga amino acids. Okay, questions so far?